Welcome to the channel. In the previous video, we briefly looked at how the cell cycle is regulated via cyclins and cyclin-dependent kinases. Now let us look in detail at how the G1 phase is regulated. So here we have a cell in the early G1 phase. This cell receives signals in the form of growth factors from neighboring cells. These growth factors bind to growth factor receptors on the cell, activating the RAS, RAF, MEK, ERK signaling pathway and leading to cyclin D transcription and ultimately cyclin D synthesis. This cyclin D can then bind to and activate CDK4 or 6, allowing the cell to progress through the G1 phase. Now, if the cell does not receive these growth signals during the G1 phase, cyclin D synthesis would not have happened and CDK4 and 6 would remain inactivated. This can then lead to cell cycle arrest at G1. If this continues, the cell can even enter a specialized resting phase called the G0 phase. Now let us look at how cyclin D and CDK4 or 6 drive the cell cycle forward through the G1 phase. So let's look at our cell here. The cell has some transcription factors called the E2F family of transcription factors. These are generally bound by a protein called the retinoblastoma protein or RB. So in this bound state, the E2F cannot perform its function. Now when the cell receives growth signals, cyclin D synthesis happens, which activates CDK4 or 6. This activated CDK will phosphorylate RB, causing E2F to dissociate from RB and move into the nucleus. E2F can now activate the transcription of genes required for cell cycle progression. One protein coded by these genes is cyclin E, which can activate CDK2 to drive cell cycle progression through the start transition and G1S transition. So how is cyclin E CDK2 regulated? To understand this, let us look at our cell in the early G1 phase. At this stage, the cyclin E CDK2 complex is bound by P27, which is a CDK inhibitor. Now let us see how this inhibition by P27 is removed. So when the cell receives growth signals and the RAS, RAF, MEK, ERK signaling pathway is activated, P27 transcription is repressed. This leads to a reduction in P27 synthesis. We have also seen previously that these growth signals also lead to increased cyclin D synthesis. One thing that happens with increased cyclin D synthesis is that more and more P27 will bind to cyclin D CDK4-6 complexes instead of the cyclin E CDK2 complexes. So this will allow for some cyclin E CDK2 to not be bound by P27 and thereby allowing these CDK2s to be active. This activated CDK2 can then phosphorylate a P27 bound to another cyclin E CDK2 complex. This allows for this P27 to be targeted for ubiquitination, meaning ubiquitin molecules are attached to the P27, allowing for this P27 to be further targeted for degradation by a proteasome. So in summary, there are three ways in which the inhibition by P27 is removed. Firstly, growth signals received by the cell lead to reduced P27 transcription and hence reduced P27 levels. Secondly, the growth signals also lead to increased cyclin D synthesis, which would lead to increased binding of P27 by cyclin D CDK4-6 complexes, sequestering the P27 from binding to cyclin E CDK2. And finally, activated CDK2 can phosphorylate P27, thereby targeting P27 for ubiquitination and finally degradation. So now that we have enough activated CDK2s, how do they initiate the S phase? To understand this, let us look at the DNA in the G1 phase. So here we have the origin of replication. This is a portion of DNA from where DNA replication begins. This is bound by a protein complex called the origin recognition complex or the ORC, which serves as a platform for the assembly of other proteins required to initiate DNA replication. So we have some helicase loader proteins that recognize the ORC and load a pair of inactive helicases to the origin. Now, during the S phase, the activated CDK facilitates the recruitment of certain initiator proteins, which will eventually lead to the activation of DNA helicase. So now DNA helicases can unwind the DNA and DNA replication can proceed. And with that, the cell can duplicate its chromosomes in preparation for the next phase that is to come. And that's it for this video. If you found this video helpful, do like the video and subscribe to the channel for more such videos. Thank you.